Don Haas, the Director of Teacher Programming at the Center for Climate Change Education at the Paleontological Research Institution and the host of Science in the Virtual Pub. And this is our final session for the spring of 2025, and we just had our 5-year anniversary, and you can read a blog on that there. And tonight's session is Charismatic Microflora, introducing the hidden freshwater algae all around you, with Dr. Sarah Whorley from Daemen University. And I am going to stop sharing my screen. I'll give you another moment to hit that donate, but there we go. And take it away, Sarah.
All right, I will transition here. All right. Just one more double check that you're all seeing the correct actual presenting screen and not presenter mode.
Yes, indeed. Looking good.
Excellent. All right. So, thank you all for coming. I'm thrilled to talk to you all about the wonders of algae. Within the biological world, small microscopic things often get overshadowed by their more charismatic megafauna, megaflora. But I think these little guys are pretty charismatic themselves. So who am I? So I am the kind of biologist that I am. I would call myself a phycologist. And what is a phycologist? A phycologist is somebody who studies algae. Phycologists come in all flavors, from the ecological to the purely taxonomic to the molecular and everything in between. I tend to fall more on the ecological side of the phycology spectrum. And so I also do double duty as a limnologist. And this was a definition of a limnologist that was hanging in my grad school lab that I just really loved. And it made a lot of sense and was kind of fun at the time. But now that I'm sort of a winter limnologist, it's extra cheeky. Here at Daemen, I do teach a variety of courses that don't directly focus on algae, but algae usually play a role in some way, shape, or form. Courses like limnology, which is just the fancy word for freshwater ecology, environmental chemistry, ecotoxicology, biostatistics, and hopefully soon some GIS, some geographic information systems. When I'm not teaching, the kind of research that I do with algae is really focused on how anthropogenic disturbance or human actions on land affect the algal biodiversity and biochemistry in local waterways, especially streams. And just because, people are allowed to be interested in things other than algae. I also supervise a lot of research students that have interests in other aquatic things that aren't algae, like fish or macroinvertebrates in streams, and any of the sort of associated and related topics that they might be interested in with those critters. So how does one become a phycologist? Because there are not too many little kids that lay awake at night dreaming of the one day when they will finally be a world-renowned phycologist who studies algae. So how did this happen? So like most of us, I went to college. I went to undergrad. I started out at the University of California, Riverside, where I'm from, where I was going for my bachelor's in biology, and I decided to pick up a botany minor because why not? I spent a lot of time working in a plant molecular lab as a work study student. So I started washing dishes when I was a sophomore. And by the time I was a fifth-year senior, I was running the greenhouse and making a lot of their reagents. So I spent a lot of time doing not algae. I also became really good friends with a grad student through taking a group fitness class at the gym on campus. And through our friendship, she recruited me to be her unofficial assistant through her dissertation, where she was looking at a species of freshwater fairy shrimp, Branchionectolindali, that lives in vernal pools in arid habitats. And she was doing her dissertation trying to figure out the geographic distribution of a virus that was skewing the ratio of males to females within those isolated populations. Okay, so none of this is algae. So how did we get there? Well, when I was an undergrad, in addition to doing all of this lab work, what I thought my degree was sort of working to was that I was going to go be a high school teacher. And at some point, somebody said to me, well, if you have a master's degree, they pay you more as a high school teacher. And no one bothered to tell me that that was a master's degree in education. So I applied to biology programs because I was a biology major. Clearly, I needed to go to grad school in biology. So I applied to grad school in biology, and beyond my wildest dreams, I actually got in. And I ended up going to Eastern Michigan University. So I basically threw darts at a map and was like, where else could I go that I might want to live? And Michigan sounded pretty nice. They had winter. I'd never seen winter. So I went to Michigan. And I got to Michigan and I got to my very first day of graduate school. And it was in mammalian histology, again, still not algae. And the professor thought it would be a fun idea to go around the room and everybody introduce themselves, since there were only like 12 of us. And so everyone's going around the room saying, hi, I'm so-and-so. I work in Dr. Such and Such's lab and my thesis is on blah, blah, blah. And then it got to me and I was just like, hi, I'm here. And I walked out of that very first day going, oh crap, I have screwed up royally. I'm supposed to have a thesis advisor. I'm supposed to be doing research. I am blowing my second chance at doing a good degree. And so after I went home and calmed down, went back the next day, went to that professor and was like, so I seem to have made a misunderstanding about what I'm supposed to be doing here. Do I need to do research? What is this? And he talked me off the ledge again and basically said, you know, if you want to do research, you need to go find a thesis advisor. Go. And so I just started walking up and down the hallway of my new department where I knew no one and was basically knocking on faculty doors going, will you be my thesis advisor? Because I did not know what I was doing. And thankfully, somebody took pity on me. And that person who took pity on me was a freshwater phycologist, an algae professor. And he took me under his wing and brought me into his lab and taught me everything that I needed to know to survive my master's degree. And it turns out not only did I like working in his lab, I really loved working in his lab. And I really loved studying algae. And I loved this whole idea of freshwater. Growing up in Southern California, there isn't a whole lot of freshwater. So the idea that there was just water around all the time was unique. And so that was a really fun thing to do. So that was what really got me started in algae and taught me that, you know, I wasn't a very good undergraduate. I struggled as an undergraduate, and graduate school sort of gave me that second chance to find myself as a student. But it also really taught me that I really liked doing research. I really liked that part. I loved going out in the field. I loved being in the lab. I loved all of it. And so I wanted to keep doing it. And so I decided that I needed to go get a PhD, and that's how I ended up at Fordham in New York City, working with another professor of algae there and doing my dissertation on the effects of agriculture and best management practices on algal biochemistry, and thus a phycologist is born. All right, so what is this algae stuff that I've been talking about? So mainly algae live in what we would call the kingdom of Protista. So Protista is not a real true taxonomic kingdom, but it's that sort of hodgepodge place where we don't have, where we put everything that doesn't really belong anywhere else. And so algae occupy a large swath of the protists groups, but they themselves are an extremely polyphyletic group. There's no single evolutionary origin. So here we've got a phylogenetic tree showing mainly the groups of algae. So all of the colored boxes on this tree are taxonomic groups that we would consider algae. And you can see that they are all over the place. We've got cyanobacteria over here in the bacteria domain, no algae over in the archaea yet. I'm sure we'll find some eventually. But most of our algal groups are eukaryotic, and they live all over the place. Basically, every group within the eukarya has some sort of group of algae, except for these guys, the amoebozoas and the opisthecons over here. These are the only groups in the eukaryotes that don't have any algae in them. But every other group of eukaryotes has algae. Most of the time when we think of algae, we're probably thinking of these green algae that are most closely related to land plants, but large swaths are not at all associated with land plants and are not associated with cyanobacteria. So basically, the thing that really ties all of these disparate groups together is the fact that they use chlorophyll to do photosynthesis, and at a single unit cell, they are fully functional as their individual species. If they're normally a macroscopic thing, if you isolate one cell, they're usually pretty okay with that. If they exist as one cell, then they're going to be okay as one cell. Otherwise, it's a pretty hodgepodge group, which makes them challenging, but also really interesting to study. So I study primarily the freshwater kind of algae. There are lots and lots and lots of marine algae, but I'm mostly on the freshwater side. So on the freshwater side of the algal world, we have all of those groups that I showed you before, just like on the marine side. Except here in freshwater, most of the time we are looking at something microscopic. So there aren't too many macroscopic or visible with the naked eye freshwater algae that you can see. And they're all photosynthetic in some way, shape, or form. This is an incredibly taxonomically diverse groups with many thousands of species. Diatoms alone can have over 10,000 species. So we're talking lots and lots and lots of species. It's very commonly a thing in grad school to sort of ask people what your study species is. And I always had a really hard time with this question because I don't have just one study species. I have, and on any given day, I have anywhere from 400 to 5,000 study species. There's just a lot of them. Algae are incredibly useful for lots of purposes. I personally find them incredibly useful as biological indicators of disturbance, which I'll talk about a little bit more later on. Amongst all of these different species, there are very, very few that have a common name. And amongst those that we have named, their taxonomy is changing constantly. And so you really have to kind of work at keeping up to date on their taxonomy. And learning all that taxonomy can really be a barrier to entry to getting folks into the field. So just some quick examples of what I mean by common names. So I have here the genus for several different groups here. One particular species that I like and find very charismatic is Gomphonemia angustatum. This is one of the few algae that does have a common name. It's sometimes known as the Mae West diatom, and that's because it is highly curvaceous, like the movie star Mae West. I sort of think it looks like a Barbie doll that you popped its head off of, but it does have its own common name in some circles. And regarding that changing taxonomy, I'll mention this one again a little later on, but recently, and when I say recently, I mean it's been like a 2 to 3 year process. The portions of the genus Anabina have been sectioned off and put into another genus called Delicospermum. So a lot of things that I learned maybe 15 years ago as Anabina, I now have to retranslate in my head as Delicospermum every time I see them. So there can be a lot of taxonomic changeover that makes this a challenging group of organisms to work with. As I mentioned, these are mostly microscopic. There aren't a whole lot of freshwater algae visible to the naked eye. Most marine algae that we think of, like seaweeds, are very macroscopic, like the bull kelp that can be hundreds of meters in length. But even in marine habitats, we have microscopic phytoplankton. On the freshwater side, it's mostly the microscopic things with very few occasional macroscopic things. So on the macroscopic side, things that you can see with your naked eye is this red algae Lemania. I know it doesn't look red, but it is in the red algae group. So Lemania is something that you can see with your naked eye. It sort of looks like those beaded chains that you sometimes get like your ID card on sometimes. Herobodiella is another one that you can kind of see macroscopically sometimes. Excuse me. So Herobodiella is a crust-forming kind of algae, and you will recognize it on large rocks where you'll find these sort of circles of dark, 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 dark brown, like coffee that has really solidified and stained. Here it is compared to a paint scraping razor blade. So you can see this crust is pretty small. So if you know what to look for, you can see these guys like Herobodiella. Sometimes Hydrodictyon gets to be pretty obvious. So here's a macroscopic filament of Hydrodictyon. It kind of looks like a lace tool that somebody might have dropped in the water. And when you put it under the microscope, it tends to look like this. So macroscopically, not terribly interesting, but microscopically, it gets to be really pretty. All right, so you're hooked. Now you want to go out and collect some algae. How should you go about doing that? Thankfully, the materials that you need to collect algae are pretty simple and straightforward. You can just get some tweezers and you can find some filaments and just pluck them off rocks with tweezers. My tool of preference is to use a toothbrush. just as means of a prop right here. So here is the toothbrush that I used all through grad school to collect algae off of rocks. You can see that so I spent 3 years scrubbing rocks with this toothbrush, and you can see that that really did a number on this toothbrush. But I kept it as a souvenir. But just a regular old toothbrush is a perfectly fine tool for collecting algae. You can use this turkey baster here. Turkey basters are really great for collecting some very fine algae that likes to grow on sediments. So in this picture here, I know it's kind of hard to see because the sunlight is glaring on the water, but there's these sort of powdery streaks on the sand here. And these powdery streaks are strands of filaments of diatoms. And the best way to get those without getting too much sand is to use a turkey baster. You can also order a plankton net of various mesh sizes so that you can get fine or really small or maybe larger algae, but those are probably the most expensive piece of equipment you would ever want to buy. Once you have your scraped off or plucked off or squeezed up algae, you can just chuck it in a little bag or a little bottle. You can use water bottles, you can use Ziploc baggies, whatever you've got on hand. It doesn't need to be anything special. Once you've got your algae, how are you going to see it? Unlike other photosynthetic fields like botany, where a hand lens might be your tool of preference, that hand lens isn't going to get you terribly far because, again, these things are microscopic. So a lot of phycologists eventually get to the point where they're ready to invest in a field microscope. These can be on the expensive side, but they are probably worth the cost. Two sorts of field microscopes that are currently available on the market that are fairly reasonably priced are the IO light microscope, which is this guy right here. So this one is really neat because it uses Bluetooth to talk to your cell phone. So you just slap a slide down on it. This is the lens, this is the slide, and then this part down here is the light projector part, and then it just sends the image right to your phone. The only problem with that is that if you are like out in the boonies, the Bluetooth is going to be terrible. And so if you're really, really remote, this might have trouble communicating. And so for that reason, my personal preference for field microscope is this EM microscope that sort of looks like a little flying saucer, but it does have this ocular piece that you can look at directly in the field. It even comes with an attachment so that you can put your cell phone up to it so that if you wanted to take pictures or not hold the thing right up to your face, you could do so, but it does not require Bluetooth. So I kind of like that. If you wanted to just do this at home and you were just going to throw things in a bag and then look at them later, that's fine. Standard compound microscope will get you to 90% of the way there. They're pretty cheap, used on eBay, and go up to 400 times magnification pretty easily. And so you can look at them that way. All right. So once you've got them under the microscope, what are you looking for? So first, we're going to talk about their growth form. How do algae occur? Algae occur in all varieties of cellular conformation that you can get. We have algae coming in unicellular varieties, colonial varieties, and even filamentous varieties. So as an example of a unicellular, I've got here star astrum. As an example of a filament, I've got here some desmidium. Here are some filaments of a duonella that is growing on a filament of lamania. So here's that large lamania that I showed you before. Here it is under the microscope. And then here's some other filaments growing on it. I neglected to put a colonial one up here, but Gomphanema can be kind of colonial, especially if these filament points are kind of close together. They'll sort of get this starburst daisy pattern going on. Algae tend to grow either upright, like on these stalks, or they tend to grow sort of pressed down onto a rock so that as water is flowing over them, it doesn't just blow them away. More planktonic varieties like the star astrum, they've got all these projections all over them that act kind of like the wisps of a feather to slow their descent through the water. So they're going to have all sorts of little shapes on them. They're going to be found in all sorts of places, growing on all sorts of surfaces, free floating in the water as planktonic or growing attached to some substrate as periphyton. All right, so that's kind of generally the shape that you're looking for. So as you start to look at these guys, who are we going to see under our microscope? So some really common groups of algae include, first of all, the cyanobacteria, which are also sometimes called blue-green algae. Cyanobacteria come in all the shapes, unicellular, colonial, filamentous. Cyanobacteria are pretty distinct in their filament nature because they do exhibit this pattern called false branching. So normally when something branches at the branch point, that cell will sort of have a little fork in the road. But cyanobacteria, when they branch, it's actually two cells side by side going one way and then the other. The cells don't split themselves into a little Y shape like other things do. And when you get the cell really close up, you will notice that the cells appear uniform in consistency because they are prokaryotic. They have no organelles. So here, for example, we've got some Crococcus, and you can see that the texture throughout this cell here and throughout this cell here is pretty uniform and consistent. Yes, there's some granulation, but that granulation is evenly distributed throughout the cell. No weird lumps to one side or the other. It's all even. That's pretty charismatic of cyanobacteria. In terms of what colors you're looking for, so cyanobacteria primarily have chlorophyll A. That's the only chlorophyll that they have. And they also have some accessory pigments, most notably phycocyanin, so kind of a reddish purpley kind of. So cyanobacteria, despite their blue-green name, will appear sort of the whole spectrum of colors. They will be everything from dark green, light green, black, brown, red, purple, pink, all the colors. So here's this kind of forest green Crococcus. Here's more of an olive green Aphanizomenon being filamentous, and then almost brown-black Microcystis up here. When you are finding cyanobacteria, what does it mean that you have found cyanobacteria in your sample? Well, it can mean a lot of different things. It really depends on what species you're finding or what genera you are finding. Lots of cyanobacteria genera and taxa can produce some toxins. So for example, this Microcystis up here Microcystis is very well known for producing the toxin microcystin. Dolichospermum is known for producing some antitoxin, and other cyanobacteria are going to produce other toxins. If you're finding a lot of cyanobacteria, a high proportion, then that can tend to suggest that you might have some low water quality issues. In terms of how cyanobacteria sort of are interacting with our food webs, they're primary producers. They're going to be, doing a lot of photosynthesis, pumping out oxygen, turning carbon dioxide into sugars. But nobody else in the food web really eats them. There's not a whole lot of predators of cyanobacteria. So if you have a lot of cyanobacteria, that means that you really don't have a lot of food for your higher trophic levels. So for your fish or for your insects or for your zooplankton, there's just nothing really for them to eat if it's all cyanobacteria. All right, another group that you are likely to find under your microscope include the green algae, the chlorophytes. So this is that group of algae that's most closely related to land plants. They again come in all the shapes and sizes. They can also have additional structures like flagella. In the case of Chlamydomonas, they can produce a lot of mucilage. They can make these sort of bubbles that hold themselves together. So for example, this Sphaerocystis right here. So this is a colonial type of algae, and it'll have these sort of individual colonies, and then often the colonies will be all glued together with mucilage to make a sphere of Sphaerocystis little colonies. Something here like Bulboceti, you'll have these nice sort of bulbous cells, and then they'll have this kind of prong whip coming off the distal end of every filament. A lot of times for a lot of the green algae, because now we have organelles like chloroplasts, the shape of that chloroplast is going to often be indicative of at least the genera that it's in. So for example, this Microsterias, so the shape of the cell is this snowflake pattern that you see, and the chloroplast works really hard to fill every inch of that snowflake shape. So Microsterias is notable because it has two mirror image chloroplasts. So all of this is one chloroplast and all of this is another chloroplast. Most green algae are going to appear that grass green color, that sort of Crayola green color. And that is because they are going to have a mix of chlorophyll A and chlorophyll B. So chlorophyll A is that sort of darker green. Chlorophyll B is a little bit lighter green. You mix it together, you get that Crayola grass green. Depending on the exact taxa you have, you might have some other accessory pigments like some xanthophylls that give you something a little bit more yellowy or something a little bit more carotenoidy, right? You can sort of get a mix of colors, but they're usually in that bright green planty sort of color. In terms of what it means that you're finding green algae in your sample, these are moderate water quality indicators. These are basically the salad, right? So if imagine you're going to a salad bar, right? When you get that salad, you're first getting your base lettuce, your romaine, maybe your spring mix. And if that was all you were going to eat, just that lettuce, sure, you might get full, and it's not devoid of nutrients, but it's not what we would call a complete meal, especially not for us. So having green algae is a good base, it's a good start, but you always need a little bit more when we're thinking about our higher trophic levels that are going to be consuming these guys. And no group is without their downsides. So there are some green algae that can be considered nuisance forming. So here in New York State, the DEC can consider Cladophora in some circumstances to be a nuisance algae type, especially when it's growing in really thick mats that people don't want to go swimming in, that they're having to clean off their boats, that's mucking up their docks. So it can be a problem. All right, and then our last big taxonomic group that we'll talk about are the diatoms. So diatoms are in the group of algae known as the bacilliariophytes. And just like everybody else, diatoms come in all the shapes, unicellular, colonial, and filamentous. And diatoms are especially notable for several reasons. First of all, they are highly symmetric organisms, meaning that they're always going to have at least one plane of symmetry. So for example, this star Aeneas is going to have two planes of symmetry. You can cut it this way, and you can cut it this way, and it will be a mirror image organism. This epithemia, on the other hand, only has one plane of symmetry, this way. This is the only way you can cut it in half such that you get that mirror image. They're also going to be very notable because diatoms are a group of algae that produce a structure known as a freshwater, and that freshwater is going to be made out of silica, basically glass. So these are algae that secrete little glass jewelry boxes around themselves. And so this star Aeneas example and this epithemia example, these are actually completely dead cells. And what you do to get these images is you have to take your diatoms and you have to boil them in acid. When you boil them in acid, that cleans out all of the cellular guts, so you're left with just these beautiful glass jewelry boxes of cell casings that they were in. This Melosira down here, this is a non-cleaned sample. This is a living sample, and you can see, for example, all those chloroplasts in there. So all of these sort of blotchy yellow shapes, those are all individual chloroplasts. So this Melosira is living, and you notice that when it's living, when all of that gunk is in there, you can't see all of the pretty structures that glass has been making. So diatoms are always best viewed, especially if you want to get down to species level with the identification. You've got to clean them by boiling them in acid, and then you want to mount them in a really good microscopic resin. And then you need to view them at an extremely high magnification, often 1,000 times magnification. Most standard microscopes only go up to about 400, so you do need a special lens for this. And so you do need a little bit of training in order to view these guys well. This yellowy color comes to us courtesy of chlorophyll A and chlorophyll C, right? So we're sort of getting lighter in our chlorophylls as we're going up. So chlorophyll A was that really dark green, chlorophyll B is that lighter green, and then chlorophyll C is even lighter green. And we often get a lot of carotenoid and xanthophyll accessory pigments that give diatoms this sort of golden, yellowish, brownish sort of color. Generally speaking, diatoms are good food web water quality indicators. So diatoms are pretty slow growing compared to the other groups. And most notably, as their primary photosynthetic product, they produce lipids instead of sugar. So this is not to say that they don't produce any sugars, but they're oftentimes going to be taking those sugars apart once they get them and then reforming them into lipids. So these guys are, if we're going back to our salad analogy, right? So we've got our base of green algae lettuce, and now we're going to start putting some nuts in there and some bacon bits in there and some cranberries in there, and we're going to put some dressing all over it, right? And that's where we're getting the bulk of our calories. That's where we're getting the bulk of our nutrients and nutritional quality, is from those stuff that we put on the green salad. And this is the stuff that food webs consider the stuff you put on your food web salad. So generally speaking, diatoms are pretty good water quality indicators. There is a downside to everything, and so we can't ignore the downside that some diatoms can have. So most notably, there are two diatom species that tend to be problematic. In North America, we tend to have some problems with Didymosphenia geminata. I have a picture of Didymo somewhere else, so we'll see Didymo later. But Didymo is a bloom-forming diatom that really likes low nutrient streams. It's the weirdest bloom-forming diatom you've ever seen. So that can definitely be a problem. The Esopus Creek down in the Catskills has a rather large Didymo problem that the New York State DEC is constantly working to manage. On the marine side of things, there is a diatom that can cause blooms and can cause a lot of problems, and that's Pseudonychia. So Pseudonychia is a big problem in the northern central Pacific, where it produces a substance called domoic acid that is known to cause a lot of neuro problems for cetaceans like seals, sea lions, walruses. If there's a Pseudonychia bloom going on, then we start to see a lot of problems with whales beaching themselves. Or not whales, I mean seals and sea lions, those other things. All right. Within our diatoms, diatoms always tend to get the most attention out of all the microscopic algae, and that is because, again, of their glass shells. So if you are really getting into diatoms, then you're going to want to first learn how to identify some of the traits about them. So basically, all diatoms come in two basic shape forms, pennate or centric. So here we've got some Gomphonema. This is an example of a pennate. Here's Didymosphenia. It is another example of a pennate. If I go back, this Melocyra, this is an example of a centric. So how do I know if something is a centric or a pennate? Centric or pennate means that when you look at the top face, so if we look at our diatom here, the top face of this glass structure, this is called the valve face of the freshwater. So pennate versus centric comes down to what is the plane of symmetry on your valve face. If there is one sort of long axis, then we tend to call it pennate. Even if there's all sorts of shapes and curves to it, it tends to be pennate. If I go back to this Melocyra, this is the valve face, and we can see that it's very radial in its symmetry, all sorts of planes of symmetry. So this means that it's going to be radial. The other sides of the diatoms, if you're not a valve, then you are in the girdle range. So girdles, these are these bands of silica that sort of stack up and make the cell extra thick. So when you're looking at a diatom, it's useful to know what both the valve and girdle form look like. So here's Gomphonema looking on its valve, basically looking at its face. And then here's the girdle form, basically looking at its side. Once you've identified that you're looking at the valve or the girdle, then you want to start looking for various shapes, things like striae, punctae, and costae. These are all different sorts of shapes and markings that the diatoms can make on their frustules. You also want to note if your diatom has a structure called a wraith. So a wraith will be this line that's usually down the center of at least one valve if your diatom has it at all. And what that wraith is for is it's for moving around. So here are some Navicula diatoms using their wraith to move around. All right. Sorry, I realize I'm going very fast, but okay. So if you're not a cyanobacteria, a green algae, or a diatom, what other kind of algae are you? Again, there's lots of different types, but some other common groups that you're likely to encounter include the rhodophytes, the red algae, the chrysophytes, the golden algae, the dinoflagellates, and the euglenoids. So here's just some quick examples of a rhodophyte, Paludicola, a chrysophyte, Hydruus, and a euglena, Phacus. I didn't have room on here to cram in a dinoflagellate as well. We could, again, go through all of those main characteristics for all of these groups. They all have similarities. They have some differences, but these are all also examples of groups that you might see. All right, so it also matters when you're going to go collect your algae, because not all of the algae are available all the time. There's going to be some seasonality to this, just like not all plants are available at all times of the year. Not all algae are available at all times of the year. So there is a lot of seasonality to when these things are likely to occur. Basically, diatoms like it really cold. If you're not in a cold habitat, then you're probably not going to find a whole lot of diatoms. So diatoms tend to do really well in mountain streams where it's really cold. They do well in snowmelt, and they do well in wintertime. So wintertime, cold times, that's when you're likely to have your diatoms. Green algae like it sort of moderate. So green algae like it warm, but not too warm. They like it cool, but not too cold. And they're never going to be as many green algae as there are likely going to be diatoms. If you're on the hunt for cyanobacteria, then you generally want to keep things warm. So the warm seasons, summer seasons, that's when you're going to tend to find your cyanobacteria, both in streams, but also in lakes. All right, so those are your basic algae. So now that we know our algae, what are they good for? So I am of a group of phycologists that like to use algae for bioassessment, meaning that we like to try and learn things about what the environment is doing based on the algae that are there. Despite their microscopic nature, there is a long history of using algae for bioassessment purposes. One of the earliest papers that used algae for a bioassessment purpose goes all the way back to 1908, Colquitt and Marson, in their paper that I will not dare try to pronounce. We also have a really long history here within the United States of using algae as a bioassessment tool. One of the founders of basically the modern field of algal bioassessment was Dr. Ruth Patrick. She was a limnologist at the Stroud at Drexel University in Philadelphia, and has a whole institute named for her now at Stroud in Pennsylvania. But she basically wrote the field in how you use algae and specifically diatoms for the purposes of bioassessment. There are lots of manuals and books and guides on how to use algae in bioassessment. One of my favorites is Algae in Water Samples from 1962. This is a book that's out of print now. One of the things that happens to you when you get really into algae is you start to become a collector of old books. And so this is one of those old books that you can find. It's not terribly expensive if you can find it. And the thing that I like about it is it has these really beautifully hand-drawn plates of the different kinds of algae. This is sort of really great also for seeing the changes in taxonomy that have happened, the names that we used to call things way back when and how those names have changed now. The names may have changed, but the meanings of those algal presences have not changed. And one of the things that I really like about this book is that it really shows you that it's not the bioassessment of water quality based on algae is never down to just one species. There's never going to be one species that you find and you can go, aha, that's it. This water is perfectly healthy, or aha, that's it. This water is perfectly screwed, right? That's never going to happen because, so for example, this guy right here and this guy right here, if you could zoom in and see those names, you would see that it's the same name. They're drawn differently for reasons. Couldn't tell you what those reasons are, but the name attached to those drawings is the same name. So that name is the same. What else do we have that is the same? We've got this guy and some of these other guys are fairly closely related. Oh, here we go. We've got this guy and this guy. Those are also the same, right? So just the presence of one species is never enough. You always have to look at the composite, the entire composition, all of the different species present. So what do we do? You know, how do we learn about the environment by taking algal samples? Well, we do this in a very similar way to how we learn about human health. So when you are sick and you go to the doctor and the doctor wants to learn about what's wrong with you, what does he do? He takes a sample. And depending on what he's doing, he might take several different samples. He might take some blood cells. He might take some serum. He might look at parts of your anatomy. He might look at how your liver enzymes are interacting with your digestive system, right? He's going to look at all of the interactions. The doctor will look at the interactions, your anatomy, your fluids, your cells. We do the same thing in the environment. From the environment, we are going to take cells and we're going to look at those cells. We're going to look at the various fluids that are in the cells and outside of the cells. We're going to look at the anatomy of the organisms. Are the organisms mutated in some way? Are they differently shaped? Are they small? Are they large? What are they looking like? How are they interacting with each other? Is this one species eating this other species, which it normally doesn't do? So we use algae in a lot of the same way that we would think about sort of sampling and learning about our own health, but just for a whole ecosystem. Some of the things that I'm looking for when I do algal bioassessment is I am first and foremost looking at taxonomy. who are the species that are there. We can do that for all of the different kinds of algae that exist. But a lot of our really good metrics for saying yes, good, no, bad involve diatoms and the ratios of the different kinds of diatoms present. We are looking at biomass. How much algae is there? We can assess biomass with both chlorophyll, usually chlorophyll A, but we can look at the other chlorophylls as well. And we can also just get a rough biomass with some ash-free dry mass. We can look at the stoichiometry of those algae. How much carbon do they have? How much nitrogen? What are the ratios of carbon to nitrogen, carbon to phosphorus, nitrogen to phosphorus? And we can look at really specific molecules like lipids and stable isotopes. And lipids are actually one of the key molecules that I'm really interested in. So when I say lipids, I'm specifically looking at fatty acids. So these are these long-tailed hydrocarbon chains. These are, if you think back to maybe intro bio, where you were learning about what makes up a cell, and you learned about the phospholipid bilayer, right? So these are the two sticks of the lollipop that come out of your phospholipids to make your phospholipid bilayer in a cell. And the different lengths of these fatty acids in that cell membrane and in other cellular context are used for all sorts of different things. When we're looking at fatty acids, we are primarily interested in two classes of molecules, the omega-3s and the omega-6s. So what makes a fatty acid omega-3 or omega-6? It all depends on where the last double bond is. So here's an example of an omega-3 fatty acid. So the omega end is the furthest from the carboxyl end. So this is the omega end, and we go from 1 carbon to carbon 3 carbon, and that's where our final double bond is. So this would be an omega-3 fatty acid, and it is in fact icosapentaenoic fatty acid, or EPA. On the omega-6 side of things, we would go again to the omega end, 1, 2, 3, 4, 5, 6. That's where our last double bond is. So this is an omega-6 fatty acid, and this is arachidonic acid. The omega-3s tend to be classified or thought of as good fatty acids. So icosapentaenoic and the elongated form of EPA, docosahexaenoic or DHA, these are very good fatty acids. So if you, the next time you go to the grocery store, if you look at a can of tuna or maybe a package of salmon, you will see on that label, they proudly declare that this is a omega-3 rich food high in EPA and DHA. This is the EPA and DHA that they are referring to. And those fatty acids are really important in overall cellular function, mitosis, especially neural development. DHA is really important in neural development. And fish tend to be really high in especially DHA. Now, fish themselves can't actually make DHA. They are getting it from algae. They get it from the algae who were eaten by the bugs who were eaten by the fish. And so that's why fish often gets declared as brain food, and why pregnant women are encouraged to eat a little bit more salmon than they might normally eat, because it helps with brain development. On the other end of things, omega-6s are also important, and omega-6s tend to be very important in stress signaling pathways. The arachidonic pathway is a really important inflammation signal pathway within our bodies. And so it's important to have plenty of omega-6s around, because you need to indicate when things are going haywire and turn on the appropriate cellular signal pathways. But you can have too much of a good thing. And if you have too much omega-6s, then that might mean that your inflammation cycle is turned on all the time when it shouldn't be. So looking at how the proportions of our omega-3s and omega-6s within our algae, within our food web, might be affecting those higher trophic levels is a part of what I do within my bioassessment work. So when you go out to do bioassessment, you can do that one of two ways. You can do it qualitatively or quantitatively. When you are doing things qualitatively, you're generally just sort of looking for who's there, and you might want to know some metrics about those guys, but mainly you're just sort of looking for biodiversity. So the general purpose of qualitative bioassessment is to get an overall general sense of biodiversity. When we do things quantitatively, we usually take a few more measurements associated with our collections, and we are ultimately aiming to translate that biodiversity and biochemistry to ecosystem function and overall health. One of the main ways that we encounter algal bioassessment on a regular basis in our society is through the monitoring of harmful algal blooms. So here is the western basin of Lake Erie, for example. Western basin of Lake Erie is famous for its microcystis blooms. So this is all microcystis blooming. It is visible from space. Microcystis here in Lake Erie sometimes produces that toxin, sometimes it doesn't. And a large part of the bioassessment is trying to figure out whether it's going to produce that toxin or not. There are other places where other kinds of algae are forming harmful algal blooms. So down in Florida, they have a lot of dinoflagellate blooms, namely with the Carenia brevis dinoflagellate. I talked already about the Pseudonychia blooming in the Western Pacific, or sorry, the Northern Pacific on the West Coast. And within our freshwater systems, we're often looking for those cyanobacteria, like a phanozomenon, or sorry, like microcystis. Here's microcystis listed here. But usually sort of the trio, the nasty trio of cyanobacteria that cause harmful algal blooms include Anabina, which we now know and we now call as Delicospermum, a phanozomenon, which I showed you, and microcystis. And they often go by the moniker Annie, Fanny, and Mike. And just to show you the difference now between Anabina and Delicospermum, so here is something that we would definitely call Anabina, and you can see how uniform all these cells are. They're just sort of a continuous green. Whereas this Delicospermum, it almost looks like it's sparkling, like they're little gemstones. They are uniform in their sparkling, but they are very mottled compared to the Anabina. And so that's one of the things that we now consider a distinguishing factor. Amongst the research that I do at Daemen with algae and bioassessment, I do work with algal blooms occasionally. So one algal bloom that we actually just had published just earlier in April, actually, is about a Euglena bloom. So for a long time, it was this bloom in Ellicott Creek was just assumed to be another microcystis bloom. Someone finally threw it under the microscope and went, hey, that's not microcystis, that's something else. And they sent it over to me to work with, and I put a student on the case, and this undergraduate student has now published a paper working on this Euglena bloom. So if anyone is interested in that, I can send that paper along. Other bioassessment work that I do here at Daemen using algae includes looking at, again, those fatty acid and lipid compounds and how winter road salt application affects them. And so this is, again, another project that was started by a student. So this is Cassie. She started working on this project with me in 2018. And her senior thesis work also led to a publication where she went out with me and we collected algae all throughout the winter, and we looked at those lipids, and we found that it was a pretty close to significant, close enough for me to call it interesting, relationship between increasing chloride concentration in the water and an increasing amount of omega-6 fatty acids. So you remember I said omega-6s are indicative of potential inflammation pathways, which, algae might not experience inflammation, but if you eat a lot of omega-6s, you might. And so those food webs might be being affected by this increasing salinity, increasing chloride concentration from road salt, and having their food quality lowered because of it. And it was Cassie's study that led us to the current project that we are just now wrapping up. And so we're just, next week, we're going to be finishing a full year of regular sampling, not only here in Western New York, but also in the Catskills and also downstate as well, where we have been measuring the salinity of the water continuously for a whole year and doing monthly algal and macroinvertebrate collections of that water. To look at how road salt application going, all throughout the growing season and then into winter and then coming back out of the winter into the growing season, how those communities are being affected by increasing road salt salinity. So we have these nice hobo loggers to make those salinities, and we are measuring all these things. We were lucky enough to have this study funded by the USGS and a little bit of extra pocket change from Erie County and Niagara County. And we are doing our best to catalog all of our findings at a website to track all of this work. And the EPA has even already started using the water chemistry that we've been collecting already to make recommendations about phosphorus loading into Lake Ontario. So it's been a really big project, and I'm thankful to have had the opportunity to do it, but also really glad to not have to go out in the field every single month for a little bit. All right, so now that you're hooked, now that I've made the pitch that you are now psyched to go and study algae, where can you go and learn more? I know I'm kind of getting short or long on time here, so I'll just sort of rush through these and share my slides if anybody wants them. But there are lots of places where you can go and learn more about algae. Lots of these are often summer courses, so they're 2 or 3 weeks. They're often in a very beautiful remote location, and you can just sort of fully immerse yourself in algal knowledge. Don't have the time to go take a course, there's lots of books available. The Bible for freshwater algae in North America is this guy right here. This one definitely costs a pretty penny. If you don't have $300 laying around, then I highly recommend Dillard's as a pretty good book for learning about algae. And of course, there's lots of websites where you can learn all about algae as well. Through diatoms.org, there is actually even a certification in diatom taxonomic identification run in partnership with the Society for Freshwater Science. There's lots of challenges, as you can imagine, to algal knowledge and algal learning and becoming a phycologist. Lots of undergraduate institutions are shifting their biological curriculum to be more medical and less ecological, and we tend not to have terribly many direct medical applications of algae. I'm not saying there aren't any, but it's usually tangential rather than direct. There's also been a really big shift in ecology to using molecular tools to identify species rather than morphological or visual traits. And so lots of people are learning about algae by just sort of blitzing everything and throwing it on a PCR and getting DNA sequences for identification rather than understanding what they look like under the microscope and maybe comparing that. And we're starting to sort of come back from that molecular work a little bit. But there's also a lot of demographic changes that are reducing, like myself, faculty ability to teach algal taxonomy, right? I teach all of these other classes, but I don't have a whole lot of time within them to actually teach about algae. Once you've been trained, though, or if you're in the process of getting trained, we do highly encourage you to join one of the many algal research societies that exist out there. Here in North America, we have the Phycological Society is our the big tent that everybody lives in, and then there's usually smaller regional ones. So, for example, the Northeast Algal Society, of which I'm a member. There's also plenty of phycological work going on overseas in Europe and Asia and Africa and all over the place. And so with that, I will wrap up, and I will thank all of my students over all the years for all their hard work and providing so many wonderful opportunities to work with algae in so many different capacities. And I will thank you all for paying attention, and I am happy to entertain any questions if there's any time left.
Awesome. Thank you very much. That was great. And I'm going to open it up for questions. And you can use the raise your hand function or put things directly in the chat. And I can see Rob has put a link for his students in the chat. And I'll start off with a question.
Sure.
The way we ended up connecting for me asking you to do this was, I think, a post on either Facebook or Blue Sky or something, connected to the road salt, with some opportunities for citizen science, but it sounds like that project's done, or finishing.
Yes.
But I was wondering if you could just say a little bit about the citizen science project of citizen science aspect of that work.
Well, so I think the main citizen science part that you were aware of on probably Facebook, if anywhere, was so here in Western New York, a lot of our waterways do not have USGS stream gauges on them. This is very different compared to the Catskills, the Adirondacks, and the Hudson Valley area, where there are maybe more, ecologically infrastructure important areas like in the Catskills and Adirondacks, or just more population to worry about in the Hudson Valley. And so they've got stream gauges out the wazoo on all the streams to monitor whether things are going to flood. Is it going to shut down a roadway? Is it going to contaminate a reservoir? Things like that. And we just don't have that much ecological infrastructure and comparatively fewer people that will be affected by roadway flooding from rivers. And so we just don't have a lot of stream gauges. And I can't go out to my sites if the rivers are flooded. One, because it scrubs off all the algae, right? So all of the sediment that gets washed into these streams with big rainstorms acts like a Brillo pad and scrubs all the algae off the rock. And two, it's just literally dangerous and will kill you. Right? If you try to, there's one of my streams that when it floods, it goes from being about a foot deep to being about 6 feet deep. And that's a lot of water moving really, really fast. And it will just literally kill you. So, so I was trying to find people close to where my sites were who could report back on what the stream conditions were, because what we might see as, it's just a little bit of rain, depending on the stream, it can turn into a great big flood very quickly.
Hmm.
And so I was trying to find out, are my streams flooded? Can I go?
Okay, cool. And there's a simple request in the chat to go back a couple of slides and show the Daemen URL. And that's from Nicole, who notes she's a Western
Oh,
New Yorker, so especially interested in that work.
this one. This one?
Yeah.
Okay.
And maybe you can just copy that link off the slide and put it in the chat. Maybe.
Maybe. We'll see how good at the computers I am.
Other questions? And Velma says hi. And Rob is raising his hand. Go ahead, Rob.
Okay. There's someone at Cornell named Marsh who's working on environmental DNA to look at the diversity of freshwater algae in places in upstate New York and the Great Lakes. And I wondered if, does that form a part of any of your work, or is that too difficult to narrow down to particular species to be useful for the kind of biomonitoring that you tend to do?
Um, so a number of things. So first of all, okay, so huge caveat. I am not a molecular biologist. I have never extracted DNA in my life. I have never run a gel in my life. So molecular things are very much outside of my wheelhouse. But from what I understand from those who do molecular work, within the repositories of DNA sequences that are tied to individual species, those repositories are mainly focused on diatoms, mainly focused on cyanobacteria, some green algae, scattering of everybody else. And so it's really hard to do eDNA work and get a true sense of biodiversity, because a lot of times you can't tie those DNA fragments back to a taxonomic name. Sure, you can get, oh, there's 10,000 unique codes here, but you have no idea who maybe 40% of those codes are, or 60% of those codes are, depending on what species they are. And a lot of times, sometimes, depending on how long ago that code was entered, the name might have changed. If someone isn't sort of familiar with the ever-changing landscape of algal taxonomic identities, they might not know to go to AlgaeBase and clarify the name, make sure it's the current name. So I'm not saying that there's no use in eDNA for algal work, but it is still very early days for getting a true broad sense of the taxonomy present. And the second thing I would say to that is that once that Rubicon has been crossed and we can get good eDNA to taxonomic identifications made like that, that would probably make my my work with the biochemistry a little bit easier to sort of tie to those species, because a lot of times people want to know, like, what are the species that are contributing this fatty acid, that fatty acid, or the other. And so right now we're just sort of able to get kind of broad taxonomic groups to connect taxonomy to the biochemistry markers.
Got it. Okay. So I have a very practical then, it's not really a follow-on question, but so I'm imagining, so your students are getting practice, at least some of them are getting practice in various kinds of algal identification and doing this kind of biomonitoring. Is that an an area where most are they getting jobs in those areas, or are they going into graduate school, or what's a common trajectory for students who are doing the sort of work that you described today?
A common trajectory for students who are doing this work with me is going off to medical school.
Okay.
I have had one student go off to graduate school. Cole, he went and got his master's at SUNY Oneonta, where he did his master's thesis looking at cyanobacteria in Lake Chautauqua. And so he did a lot of morphological ID of of cyanobacteria in Lake Chautauqua and also quantified sort of the amount of cells per liter that get blown in your face when you are tubing and water skiing around Lake Chautauqua. And after completing his master's thesis, he has since gone on to become the director of research at the Squam Lake facility in New Hampshire.
Oh, that's very cool.
In terms of like what jobs are out there, so in terms of jobs that are out there in algal bioassessment and monitoring, most of those jobs are government jobs. So city, county, state, federal bioassessment. So among the people who go and take these classes. So a lot of these classes are very often staffed by various state, federal, et cetera, water quality monitors. So this is the year that I took the class, and pretty much everyone who was in this class, except for me and like one other person, worked for New York State, New York City, Vermont, and I want to say there was maybe somebody from Pennsylvania. And they all worked on the reservoirs for those various states and monitoring the reservoir water quality to make sure that the reservoirs were safe to drink. A lot of the current work at the federal level is done by the EPA with the Office of Research and Development, the ORD. That's the office that mainly monitors the cyanobacterial blooms in our Great Lakes and other main large water bodies to monitor for microcystin production from microcystis. There are a smattering of private companies that also do this bioassessment monitoring. So basically, and they're basically doing it on behalf of the state, right? So local municipality notices a water bloom or an algae bloom in their water. They take a sample, they send it off to this private company, and the private company then does the ID work. So there's a smattering of those companies. There's two in Michigan that, off the top of my head, their names escape my name, but one's in sort of like the Holland, South Haven region, and the other's in Traverse Bay. There is I know Tetra Tech does some of this work. Tetra Tech does have a local office in Buffalo. They've got some offices elsewhere. There's a smattering of private companies that will do this work, but they are mainly focused on cyanobacteria. So they're mainly looking for Annie, Fanny, and Mike. They're looking for Microcystis, Dolichospermum annabina, and Aphanizomenon. And so a lot of times they're technicians. If it's not that, they don't know what it is. Which, that's what pays the bills. So I don't blame them.
Got it. Thank you.
Yeah, and another question that came to mind for me is, do you know if anybody's putting algae and algae species in iNaturalist?
Yes. So Carolina Fuchikova, formerly from Assumption University and now works in an administrative capacity at Cornell, runs an iNaturalist page specifically dedicated to desmids. So she's specifically working with desmid green algae to catalog their diversity. I'm sure that there are other people that are doing other groups of algae, but I do know specifically of Carolina Fuchikova doing desmids.
Cool. Rob, do you know if when did is our BioBlitz still going on right now? I don't remember the end date.
It is not. It ended about a week ago. Yeah, but we did have it for about a week.
Yeah. So we do 4 BioBlitzes a year, one in each season, and the spring one just ended.
Awesome.
So that is specifically for the Cayuga Lake watershed, but folks can take a look at our BioBlitz page if they're interested in learning more about that.
And in the Cayuga Lake region, you have Dominique Derminio at Cayuga College as your phycologist in residence.
Cool.
We're everywhere.
All right. Other questions? All right. I think it was a great session and a good closeout to the season. We will return on the 4th Thursday of September and look forward to seeing people then.
Thank you for having me. It was great.
And a nice comment in the couple of comments in the chat that you should look at there.
Thank you all. I really appreciate it.
I think we had about 90 folks at the peak.
Yeah. Thank you very much, Sarah.
Yeah, it was great.
Appreciate it.
Thank you again for having me.
Sure thing. And I'm gonna close the meeting. Everybody have a good weekend and we will
Drive safe.
see you in the fall. Bye-bye.
Okay. Bye-bye.